Hello, Mr. Rector. This is Emily, and today I'm going to talking about something about the coronavirus. And since today the number of the confirmed case in United States have already over the 112 million. So I think that all of us just we stay in the United States, then we should take some action and take some measures to protect ourselves. And I'm gonna talking about something to explain the testing process and how can we detect the virus using the real-time PCR this way. A sample is collected from parts of the body where the coronavirus gathers, such as a person's nose or throat. The sample is treated with several chemical solutions that remove substances, such as proteins and fats, and extract only the RNA present in the sample. The extract RNA is a mixture of person's own genetic material and, if present, the coronavirus RNA. The RNA is reversed and transcribed to DNA using a specific enzyme. Scientists then add additional short fragments of DNA that are complementary to specific parts of the transcribed viral DNA. These fragments attach themselves to target section of virus DNA. If the virus DNA is present in the sample, some of the added genetic fragments are for building on DNA trees during the amplification, while others are for building the DNA and adding marker labels to the strains, which are then used to detect the virus. The mixture is then placed in a RT-PCR machine. The machine cycles through temperatures that heat and cold the mixture to trigger specific chemical reactions that create new identical copies of target section of viral DNA. The cycle repeats over and over again to continue and copy the target section of the viral DNA. Each cycle doubles the previous amount. Just imagine and assume we have two copies here, and the two copies become 4, and the 4 copies become 8, and so on. A standard real-time PCR setup usually goes through 35 cycles, which means that by the end of the process, around 35 billion new copies of the section of viral DNA are created from each strand of the virus in protein, which is present in the sample. So that is the process of how the RT-PCR the machine works. Thanks for watching.